What's up, y'all? Welcome back to Purple Bear Biology, where we're exploring parts and functions of our blood. In this episode, we are going to continue that conversation with a really cool and important process called blood clotting. There are multiple steps to clotting, so let's start with the primary component of our blood that is responsible for clotting, called platelets. Technically speaking, platelets themselves are not whole cells, but rather fragments of cells found inside of our bone marrow called megakaryocytes. These cells extend portions of their membrane into the blood vessels where they break off and free float into our bloodstream. These platelets contain portions of their former cells, including small chemical granules used to recruit more platelets, mitochondria to synthesize ATP for clotting retraction, and even small protein filaments called actin and myosin that we tend to associate with muscle cells for contraction. The process of forming a clot is the result of multiple pieces of a puzzle working together to accomplish the end goal of preventing blood loss. We will break it down into 5 steps, each requiring specific conditions to activate the coagulation cascade. Tight regulation is necessary for this cascade because you do not want to form clots all of the time, only when damage has occurred to the tissues or the cells inside of the blood. I should mention for clarity that the actual coagulation cascade is much more complicated than we will discuss in this video. We will be clumping a group of variables called clotting factors together, but there's actually an intricate network of activation needed for us to develop these clots, and the absence of any of these clotting factors could lead an individual to fail to develop clots when damage occurs. So let's jump right into the steps and talk about how all this works. The very first thing that must occur to activate the clotting cascade is damage. This damage could be intrinsic, where parts of the blood or blood vessels become damaged, or extrinsic, where tissue trauma occurs, exposing surrounding tissue to the blood plasma. Depending on which of these damages occur will determine which coagulation cascade will be activated. But because we're kind of blackboxing all the initial cascade clotting factors, let's take a look at what happens when damage occurs in general. The very first reaction will be for our blood vessels to contract and narrow. This immediately reduces the surface area of the vessel, making it easier to fill the damage, and reduces blood flow so platelets can begin to adhere to the site of damage. Additionally, inflammation of the surrounding tissues applies pressure to the damaged area, working just like when we apply pressure to a wound to help it stop bleeding. The next step is for platelets to begin to adhere to the damage or exposed tissue. As platelets arrive and recognize a problem, they will release a recruitment chemical in the form of chemical granules that then call in more platelets to the area. The accumulation of platelets develops into a soft plug that begins to fill the damaged tissue. For really small amounts of damage, this may even be enough to prevent blood loss, and the cascade concludes. However, for really severe damage, this soft plug will not be enough to stop the blood loss, and the coagulation cascade will continue. Both intrinsic and extrinsic pathways will lead to a common clotting pathway. In this common pathway, there are free-floating clotting factors, synthesized primarily by the liver, that need to be activated for the cascade to continue. The ultimate end goal is to get an active form of the chemical called fibrin. To do this, clotting factors actively help prothrombin convert to thrombin, and then thrombin enables fibrinogen to be converted into fibrin. One of the most important ions needed through all of this process is calcium, demonstrating another way this cool ion is vital for our survival. When fibrin is present, it acts like a web-like network that traps platelets and red blood cells at the site of damage and forms a stable, sturdy clot. Once the clot is formed, the filaments inside the platelets cause contraction to occur, and this pulls the damaged tissue closer together to make it easier to heal the tissue. You can kind of think of this clot retraction step as a biological suture, or one of those really cool butterfly band-aids. Lastly, once the tissue is healed, the body needs a way to break down the clot and recycle the components. To do this, another chemical conversion needs to take place, converting plasminogen to plasmin. Plasmin dismantles the clot and completes the coagulation cascade through a process called thrombolysis. Now, of course, all of this is what happens when things go as planned. But what happens if clots do not obey the rules? When clots form in areas that then prevent blood flow, it's called a thrombus. These clots can even break apart and become mobile, getting lodged in smaller vessels, and this is referred to as an embolus. Both of these situations can be life-threatening because they can block blood flow and thus oxygen as well as nutrients from making it beyond the clot. This is particularly common in vessels of the legs because they have valves to help prevent blood backflow due to gravity. When this occurs, we refer to it as a deep vein thrombosis. It can also occur in the vessels to the brain, resulting in a stroke, pulmonary circulation to the lungs, or even the coronary arteries of the heart. The opposite of clots forming where they're not supposed to would be clots not being able to form at all. A condition referred to as hemophilia results from a genetic mutation on the X chromosome. This sex chromosome is passed on to the offspring and results in the inability to synthesize necessary clotting factors. Interestingly, because females actually have 2 copies of the X chromosome, if one has the mutation, the other can be used to supplement the clotting factor's synthesis. Males, on the other hand, only have 1 copy of the X chromosome because their other sex chromosome is the Y chromosome. Thus, males have an increased likelihood of expressing the disease even if their parents are just the carriers, aka they do not express the disease themselves. Well, that's it for this episode of Purple Bear Biology and this conversation about the coagulation cascade. If you found the video useful, consider liking and sharing it with others that can use it too. If you have any questions or want another video to delve deeper into the clotting factors, drop a comment in the comment section below. As always, thank you all for watching and see y'all next time.